All right, class, and welcome to our live class where we'll be going over chapter 3. And before we get started, I just wanted to kind of highlight some of the most recent announcement that I posted. Please take a look at this academic integrity and exam and quiz protocol announcement, as well as the one I just posted a couple minutes ago, the last 2 weeks in our first module. And I'm just going to go over that now. And this is being recorded just so that everyone knows about this. So we just have 2 weeks left in our first module. One thing that I was going to ask you guys, if it's okay if we start all our classes at 6 p.m. instead of 6:30. If you guys are okay with that, I might change our time a little bit. If I get a lot of kickback, we can keep it at 6:30, but I might kind of move our class time up to 6. But again, let me know how you guys feel about that in the chat if you want to, or send me a message later if you are not okay with that. As usual, here's the link to all of our class YouTube recordings on our class YouTube channel. And then this is just our schedule in lecture for the next really week and a half. So this we'll be going over chapter 3, which is the cell, and I've divided it into 2 parts because it's pretty lengthy. And then on Tuesday, we'll be doing chapter 4. And then on Thursday, you'll have your first lecture exam. And I just want to make it very clear, I'll probably be bombarding you with announcements about this, but this is a slight change. From here on out, all of your lecture exams will only be open during normal class time. And a lot of online professors do this. This would be really similar, if we were meeting in person, we would often meet, and you guys would have your class during the time. And I just saw that Emily Barrera joined us. Hi, Emily. So I'm just going to signal you out quick. She's here from the Multidisciplinary Success Center to talk a little bit about our workshops and tutoring opportunities. So I'll let you talk, Emily, so you don't have to stay on the whole time with us.
Thank you. Hi, Beth. How are you?
Good.
And awesome.
Kids.
Yeah, I know. Sorry, we're just trying to finish dinner.
That's okay.
Is it okay if I share? I was going to share the physiology workshop page.
Yeah. Do you have access to do that, Emily, or do I need to give you?
It looks like you have to give me access. Yeah.
Okay. Do you know how I do that? Let's see here. I'll just make you a co-host and see if that works.
Okay.
That's good.
Do you wish to continue? Okay, there we go. Okay. Hello. Good evening, everyone. I hope you are doing well. Do you see this page, everyone, that says physiology workshops? Yes. And you have this in your page, right, Beth, on your class?
Yes. Mm-hmm.
Okay. Yes. So as your professor said, I'm just here to go over the process for the workshops, and then let you know about our tutoring opportunities as well. Obviously, this is a very difficult course, but we do have a lot of support at the college for you. So as you see, you do have, it's called supplemental learning assistance, but there's some workshops that are part of your course, and your professor has that in her syllabus. So there's 2 options to complete these workshops. The first one is you watch the video online. And as you see, if you scroll down, these are the 5 workshop topics. So you have cardiac cycle, cellular metabolism, gas exchange and acid base, etc. Each of them have an online lecture. So that's a YouTube lecture, and it's captioned, and they're about 20 minutes. So you would watch the lecture, and then each of them also have a corresponding handout. So you can preview it, and when it's time for you to do it, you can download it. All of the handouts are in Word format. They're all accessible. I did not mean to do that. And the cool thing is they have kind of like the areas where you can type. So it's really obvious where you're supposed to put an answer versus where you're just reading information. So you would do the lecture and work on your handout. And then after that, if you can make it, we have what we're calling like a live session schedule. So you can click on that. We just put that up. So you would go to whatever topic your instructor is working on. So let's say she's working on graded and action potentials. You would look and see if you can make one of these times. And it's a Zoom session. So it's about 45 minutes. And in that session, you would bring your handout. It's okay if it's not completed, because that teacher will kind of walk you through it, go over the handout, answer questions you have about it, answer questions you have about the lecture video to make sure you understand it. And for most of the topics, they do some kind of interactive activity. So some kind of game we updated for the online format, so you can get a better understanding of the topics. The good part is for these ones, you don't have to do any signups. You just show up at this time and you click here on the Zoom link and you just go in and do your thing, ask questions. And then when you're done, you email your completed handout right here. It says submit it to this email, mdsc@lbcc.edu. It helps if you write your first and last name in physio, and then someone will just kind of eyeball it and make sure you've done it. And then they'll tell your professor that you completed that workshop. So that's one option so far. Any questions about that option from anyone? How to do that one?
Are our answers going to be corrected, or do we get graded on correct or incorrect?
That's a good question. No, that's a good one. It's not graded on incorrect or incorrect. It's the staff, honestly, who's checking it is not life science staff. It's just the general success center staff. So they just want to make sure that you've, completed all or most of it to the best of your ability. So they have the answers. So, you're not writing like nonsense, but that's a good question. It's not graded. I'm not sure if you turn it into your professor, if she would grade it, but it's more like a yes, no, like, did you complete it? But that's a good question. Yeah. I don't think I've ever heard that question in all those years. Any other questions about the first option? So watch the video, do the handout, and then email it in after you attend the session.
And someone asked Emily
Oh.
if the live sessions are mandatory.
Technically, no. As you see, when I showed the schedule, it is not the most robust schedule. I do apologize. I've been having a heck of a time of getting people. A lot of instructors who used to do this for me, including your professor, she's done the workshops. They're just too booked up with classes. I could not hire them. They are just maxed out. That's a thing with part-time teachers. And I wasn't able to hire someone. So no, they are not mandatory. But that also leads me to option 2. So we've always had this option, but I'm especially advertising it since unfortunately we don't have a lot of workshops, but if that changes, I'll let your professor know. If you're unable to attend the session, you can also make an appointment with a tutor. And this term, we're kind of slowly transitioning back to both campuses. So we still have a lot of online services at the college. Our tutoring is open Monday through Saturday, but there's also some on-campus options. So if you want to do option 2, same thing, you would still watch that YouTube lecture, do the best you can with the handout. It's okay, it doesn't have to be perfect. We know you want to ask questions, and you would click on this link. And this shows our little handy tutoring schedule. For this one, I just wanted you to note the bottom 3 tabs are for the 3 kind of locations, if you will. So the first one is online. So we'll scroll down. And if you have other classes, as you can see, our department helps with just about everything. But we're scrolling down here to physiology to see when it's available for your class. Did I go too far? Wow, we have a lot of, here we go. So Physy 1, oh my goodness. Okay, so Physy 1, we have online tutoring, as you see, Monday, Tuesday, Thursday, all day Friday, and that's it. So if any of these times work for you and you want to talk to a tutor, either about the workshop or any content, it's not just the workshop, the tutoring is here for you. We have all the information here. You can pretty much do anything. You can email, you can text this Google number from your phone, you can call, whatever you need to do, tell the person, oh, hey, I want an appointment, this Thursday, do you have 12:30 or 1? And then they'll get back to you right away and set you up and give you the Zoom link for that tutoring appointment. If you're on campus or if you're comfortable going to campus, we also have some tutoring in LAC, and some of it might overlap. Oh, maybe not. We have Tuesday and Thursday, 12 to 4. At the LAC campus, we're in the L building. That's the library building. So you go in that building and you go up the stairs and there's a center right in front of you. That's where the tutoring is at the LAC campus. Unfortunately, it does look like 4 is the latest. That's a good question. Thank you, Jacqueline. At our college, we have to hire student tutors. So our tutors are students just like you. So we are kind of working, we are working with their schedule. So in our mind, we would love to offer everything night and day for you, but we have to hire full-time students. So the bad part is it's great to give them an opportunity and have them work with people, but the bad part is we have to work with their schedule. But let me double check for PCC, but I'm sorry, yeah, it looks like that's the latest it is. And then also at the PCC campus, that's up in the EE building. So over there, if you're by the brand new parking structure, it's just really cool. If you've been at PCC and you kind of walk up the stairs and across a little kind of plankway, the EE building is over there. And I believe he has a tutor. Again, daytime hours. And it looks like Monday and Tuesday at the PCC campus. And again, here's, it looks like the same information for the appointment. So you can use the same information and just say, I want online or I want LAC or PCC. So again, you can make and it's all free. You can make an appointment for any class content questions or to go over this information for your workshops.
Thanks so much, Emily. It's nice to hear from, it seems more official when it comes from you. And I know, especially the tutoring schedule, I wanted to let them know, especially because if you guys are in other classes, the MDSC is really helpful with your other classes too. So thank you so much, Emily, for bringing all that up.
Sure, no problem. Okay, bye everyone. Have a good evening.
All right, take care, Emily. Good to see you.
Bye, you too.
All right, guys. So let's head back. Thanks for listening in on Emily. I asked her to come. She's the department chair over there. So for your workshops, if you guys have questions on those, the schedule is up. If you can't make it to one of the scheduling workshops, like she said, you can still fill out the handout and hand it in that way and by watching the video. But remember, you have to complete 3 of those workshops by the end of the semester. So thanks for letting Emily speak for a little bit. Again, it just always seems more official when you're hearing someone else besides me talking. So if you want to go back and watch any of that information, it'll be in the recording as well. So I think I ended, just so you guys are aware that you'll have to take your exam during normal class time on Thursday, September 23rd. And I realized I just asked you if we could switch class time to 6. So again, I will make it very clear when that exam will be open for you. You have one assignment due this Sunday. Yes, Dixie, question?
I'm sorry. Going back to the one that we just covered about the SI, are you gonna grade the handouts?
No, so that was a great question. Did you, I think, yeah, you asked that or someone else. So they are just, I've been doing workshops for years and they're only graded for completion. So no one is grading them for correctness. They're only grading them for completion. At the same time, you know, if you want to work with a tutor or you go to a live session, it's really helpful. It's like an extra hour of studying a difficult concept. So no, they're not graded. They're just graded on completion. If you ever want to go over the answers together, you can come see me in my office hours, or you can just send me an email and ask if you are on the right track for this stuff too. Good question. And how is Labster going for you guys? Do you guys like those labs? Maybe in the chat, just let me know how Labster is going for you. You have one more Labster lab due this Sunday, and this will take us kind of through the end of this module. You'll have a quiz on the 3 Labster labs. So keep in mind, you know, your lecture exam needs to be taken and is due on September 23rd, but all the other assignments, minus some labs, like you'll have a lab quiz and a case study are due at the last day of the module. And all of those due dates are updated in Canvas, so just stay aware on that. But how is Labster? Sometimes back in instructions, sometimes you get stuck on some. Okay, some of you love doing them. Good. Once we start doing the PhysioX, you'll notice that the PhysioX are a little bit more detailed. So a lot of times students like the Labster. A lot of information at once. Okay, thanks for letting me know. I'm just curious. Okay, so any final questions before we get started? Yeah, good question, Lindsay. So how to study for lab quizzes? Lab quizzes will be based on information from the first 3 labs. And when I ask you guys questions, like a lab quiz will be worth about 10 points, the same as your lecture quizzes are worth. So they'll still be remotely proctored through Proctorio. But the questions I ask you, I won't be asking you like the nitty gritty detail about something you learned in Labster. I try to keep the questions very like something like a very kind of broad topic that you would have at least remembered seeing. Because yeah, I know in general, the Labster labs have a lot of information. That's why we like them, because I think they get through a lot, even sometimes more than what we could even maybe get through in person. But yeah, in terms of quizzes, when do you need to purchase the book? You're probably going to want to get the book for sure by the end of this module, September 26th, because we'll start using Physio X Labs, which you'll need the access code for the MyLab and Mastering. Anna, question?
Yes, for the exam, or because I noticed on the quiz, you couldn't skip a question. Is the exam gonna be the same? Because I had difficulties with that because I tried to skip and then it completely froze. So then I had to restart the whole thing and it let me back in. But it said my time was set back, but I know it was not 25 minutes and I didn't get to complete the whole quiz.
Yeah, so the questions will be locked. Like you can't go back to a question, but it sounds like yours was actually freezing on you. Like you were having problems with the quiz itself freezing.
Yeah, because I was gonna skip a question and then I changed my mind and then it said it's waiting on Canvas and it just stood there for a while. And then I guess I was talking to the Canvas thing, the protocol thing, and it told me that it was gonna restart it for me, which it did. But like I said, it said it was gonna give me the extra time that I was sitting there for a while, but it didn't because I only got to answer 2 questions. And then it came
Bummer.
off and I was like,
Yeah, that sounds like something more like a Canvas question, but yeah, and so I'm going to keep them locked. Basically, for cheating purposes, is a big one. Also, in an online environment, just to maintain the integrity of the course. And I know you guys don't like it, but in a way, if the majority of the time when you do second guess yourself and go back, a lot of times, not always, you can pick the wrong answer. So I will keep them locked like that. If I reopen lab 2 just for you, will affect the current grade? Good question. That's a great question in the chat. I'm not sure. You can always go back and study them. That's a great question. Why don't you take a screenshot of the grade you have now, Marbelli, and then if it changes when you go back, can you send me the screenshot of your initial score? Does that help? Just so that I don't want you to get, so if any of you guys want to go back, it does keep your highest score actually. Yeah, so you're right, Emmanuel. So even if you go back and don't answer any questions, right, it'll always keep your high score. Okay, more questions. Anna, Israel. Yeah. Anna, do you still have a question?
no, I don't have another question.
Okay, so yeah, Anna, I would say I didn't really answer yours, Anna. I would say, if you have problems with that again, it almost sounds like Proctorio freezes on you or Canvas freezes on you, and let me know if that happens again.
Okay, thank you.
Yeah, and Israel, a question?
Can you please unlock the last quiz? I like to go back in and retake them personally so I can memorize everything. So is there any way you can give me access? Because I don't know if you're still giving out that quiz, but it'd be nice to get access to that so I can replenish my mind every now and then.
Yeah, no, great question. I think I put that your correct answer should be shown. Have you checked recently?
Oh yeah, I just checked yesterday. I should check again today. Okay.
Because I did, yeah, it was where I wasn't showing correct answers, but now that the due date has completely passed, yeah, let me know, Israel, even what right now, but I mean, in my Canvas, it should be, yeah, shown. It shows now. Okay, perfect. Lauren, question.
I appreciate it.
Yeah, no problem.
Yes, I had a quick question just to clarify. The lecture exam shows that it's due on the 26th on the syllabus. Is that still the same, or is it going to be open on the 23rd and due on the 23rd at midnight?
Great question, Lauren. So, and that's what I'll be bombarding you. So that's a recent change that I've made, that I've decided that we're going to take all our lecture exams just during normal class time, and they'll be due during normal class time. Yes, so that was a change that I made to kind of uphold the integrity of the course. So yeah, that's a change. The exam will only be open during normal class time from 6:30 to 8. For a normal, I think 90 minutes. And then if you're a DSPS, depending on the DSPS accommodation, sometimes it's 1.5 times or double time. So I would leave your exam open a little bit more. Good question, Lauren.
Okay, thank you.
Yeah. I feel like we should just have a day of questions. Okay. Anna and Israel, do you still have a question? I think you can put your hand down, but I'm not sure how.
Sorry, I don't know how to use Zoom. I was like struggling with the reaction. Thank you.
Nope, you're fine. I know I probably couldn't do it either. Okay, thank you guys for the questions. I really appreciate it.
I got confused
Yep.
with so much asking about the lecture exam. I ended up getting confused at the end. So I come on Zoom or I go on the proctorial?
Good question, Dixie. Yeah, and I will make this really clear, like maybe in an announcement a few days ahead of time, but you'll still be taking the exam via proctorio. So you won't come on Zoom at all. So during our normal class time, you'll just be taking the exam and you'll go to the lecture exam, you'll open it up via proctorio. So I won't be watching you, but proctorio will. I don't think Zoom and proctorio can go at the same time. I'm not sure. I should find that out. All right. The exam is not 100 questions, but it's 100 points. So some questions are worth more than one. All good questions. So your lab quiz, that's due at the end of the module, and I'll give you guys a week to do that quiz. So the only thing that you need to take during class time are the exams, lecture and lab. But the lab quiz will be based on the first 3 Labster labs. And we clarified you can go back and do those Labster labs as much as you want, and it'll always save your highest score. So if you just want to go back and just work through the questions quickly, you can go back and study them too. Will it mark it late? That's a good question, Jasmine. Those are all things I don't know. For me, I've already graded, so I'm not going to go back and regrade anything, even if I get a flag for being late. Good questions, Jasmine. Those are great questions, guys. Okay, so I'm going to move on to chapter 3. So again, these first 4 chapters, I hope, are a little bit of review from anatomy. Chapter 3 is really long, and I divide it into 2 parts. And again, how I normally do this, I'm going to try to point you guys in the direction of things that I want you to study. I can't guarantee I'm going to tell you every single thing to study, but for the most part, I take all your lecture questions from items and topics that are from these PowerPoints. Chapter 3, I'm going to divide into 2 parts, and I was hoping to get through all of part 1 tonight. So I'm not sure if we'll get through all of it, but we can always finish it up on Thursday. And then chapter 4 is on tissues, and we'll be going over that on Tuesday. And when I'm lecturing, I don't want to say I'm skipping over things or going over things faster, but it's more kind of like a highlight view, and I'm going to point you in the direction to where to study, because I don't want you to get bogged down with everything. So here's chapter 3 talking about this cell. And the first part of chapter 3 talks a lot about transport mechanisms through the cell. And this ties in really nicely with your Labster lab this week. So your Labster lab this week is on the cell membrane and transport mechanisms. And that's basically what this first half is. The second half will go into some of the organelles, and we'll go into a little bit of protein synthesis, which is also really important. Someone asked in the chat if we should just study the PowerPoints or read the textbook chapters. I would encourage you to do both. However, if you're getting bogged down by a lot of material, there's a lot of studies out there that say if you are studying a subject from multiple sources, it gets to be too much. So if you want to focus on the PowerPoints, I think you can still do okay in the class as long as you're understanding the material. Sometimes reading the textbook gives you kind of another way of describing it versus just hearing me talk, as well as, if we rush over a topic or not cover it completely, the textbook might offer a little bit more information about that. So let's talk about cells. By now, hopefully you guys know about cells. And we're going to understand why the permeability of the plasma membrane can affect treatment in disease. Cell theory talks about how the cell is the structural and functional unit of life. How well we as an organism function depends on the individual combined activities of all of our cells. The continuity of life has a cellular basis, and the structure and function of cells are complementary, meaning that the shape of the cell and specific subcellular structures that it has within it will tell us what that cell does. There you have over 250 different cell types in humans, and they differ in size, shape, and their components. And these differences between the cells will lead to their differences in functions. So just to give you an example, here are erythrocytes, red blood cells. Fibroblasts help to make up connective tissue. Epithelial cells cover surfaces. Here we have muscle cells, fat cells filled with adipose. Macrophages, these are leukocytes to help you fight off diseases, nerve cells, and then a sperm cell, the reproductive cells. So we see the diversity of cells and how the structure of the cell will determine its function. All cells have some common structures, however, even though they are different, and these are the common structures: the plasma membrane, which is the outer boundary, the cytoplasm, the intracellular fluid containing all the organelles, and the nucleus, which is the DNA containing control center. So here is hopefully a picture that you've seen before, or one like it. You should be familiar with all the organelles within the cell, what they do, really comfortable with the plasma membrane and the different ways things can enter and exit the cell. So just go back and rewatch what I just said about 10 times to know what you need to know about this chapter. We'll go over the organelles on Thursday. And tonight, we're really going to be talking about transport, or we'll start talking about transport. Extracellular materials are all of the substances outside the cells. You have different classes of extracellular fluids, like body fluids. They can be interstitial, which are cells that are submersed or bathed in fluid. The blood plasma, which is the fluid of the blood. So your blood is made up of cells, and it's also made up of plasma. And the cells are kind of suspended within that plasma. Cerebrospinal fluid is fluid surrounding the nervous system. You have cellular secretions that secrete saliva, mucus, and then the extracellular matrix is the substance that acts as glue to hold the cells together. So let's talk about the plasma membrane. Your plasma membrane acts as a barrier. It separates things outside the cell, the extracellular fluid, from everything inside the cell, the intracellular fluid. And your plasma membrane plays a very dynamic role in all cellular activity by controlling what enters and what leaves the cell. And this is important because your cells create proteins or substances that will go on and affect other parts of the body. So each cell controls what proteins leave it. It also controls what kind of things enter the cell: drugs, hormones, other things. So the plasma membrane is known as the cell membrane. I'm going to skip over or briefly cover the structure of the plasma membrane. You should know this, but I'll briefly cover it. Your plasma membrane consists of membrane lipids that form what we call a lipid bilayer. There's specialized membrane proteins going throughout it, and this is referred to as a fluid mosaic model because it's made up of many pieces. You have surface structures, glycocalyx. The membrane structures will help to hold cells in close proximity together. And this is the plasma membrane and different structures that make up the plasma membrane. So spend a little bit of time just studying the plasma membrane. And again, as we talk about transport mechanisms, we're kind of going to go over what are these structures within the plasma membrane. The big thing is the phospholipids, which have a hydrophilic or water-loving head. So the phospholipid head will always be facing the exterior and interior of the cell because it loves water. And then the fatty acid tail of the phospholipid is hydrophobic, meaning it does not like water. So the tails will face each other so that they're not touching water at all. We have glycolipids, cholesterol. This is the structure of a phospholipid. And this is a look at a plasma membrane and how these phospholipids arrange themselves in a lipid bilayer. So we have two layers of phospholipids. The heads, you can see one head here is facing the intracellular fluid, and the other head of the second half of the lipid bilayer faces the extracellular fluid. That's because the heads are water-loving, hydrophilic, and the tails face each other because they're hydrophobic. They don't like water. So the phospholipid bilayer arranges itself automatically to form this cell membrane with the heads facing the water and the tails facing the interior of the membrane. We have different membrane proteins within the cell membrane. They allow for communication. There's integral proteins or peripheral proteins. The integral proteins are just firmly inserted in the cell membrane. We'll have the transport proteins, enzymes, or other receptors. And then peripheral proteins are more loosely attached. And these can be more of your enzymes or motor proteins for shape change and also help for cell-to-cell connections. So these are different membrane proteins and their tasks that they perform. So in terms of what you need to know about the cell membrane, you should know what makes up the cell membrane. There's proteins. I don't know if I asked specific questions about the different types of membrane proteins. There's integral, peripheral. In general, I think the name of the membrane protein tells you what it does. So this is a transport membrane protein. Here's a receptor for signal transduction. So some sort of chemical messenger will bind to a receptor protein, and that will cause a series of events to happen inside the cell. We have enzymes, enzymatic activity. These are membrane proteins that may be an enzyme, which help to make chemical reactions go more quickly. Cell-to-cell recognition. So these are proteins within your plasma membranes that just help cells to recognize each other. The cell membrane proteins might help the cell to attach to the extracellular matrix. So there might be some framework or some protein fibers outside the cell, and these proteins just serve as an attachment to that extracellular matrix. Cell-to-cell joining, these are membrane proteins that may be hooked together. Some membrane proteins called cell adhesion molecules or CAMs, those might come back to haunt us in a couple slides. These will provide a temporary binding site that guides cell migration and other cell-to-cell interactions. Glycocalyx are the sugars that stick out of the cell surface. This sugar coating allows the immune system to recognize what is itself and what is non-self. A homeostatic imbalance that has to do with glycocalyx is that glycocalyx of some cancer cells, for example, can change so rapidly. So for example, if you have a cancer cell, the sugar coating surrounding the glycocalyx, it can change so rapidly so that your immune system cannot actually recognize these cancer cells as being damaged. So that's one bad thing about cancer cells of many. And as well as a mutated cell cannot be destroyed by the immune system, so it's able to replicate itself. So that's where glycocalyx can come in. Cell-to-cell junctions, again, I'll pass over this rather quickly. I know I'm not, I always keep talking. These are just ways that cells are joined together. Some cells are free, such as your blood cells, your sperm cells, but many cells come together to form tissues and organs. And there's three ways that cells can be bound together: tight junctions, desmosomes, and gap junctions. You should know generally what these are. These are integral proteins on adjacent cells that form to fuse an impermeable junction. So meaning nothing can get through, so it'll prevent fluids and most molecules from moving in between cells. So here's a look at tight junctions shown in this box, forming kind of an interlocking impermeable junction between cells. Desmosomes, they kind of interlock like the teeth of a zipper. They form a button-like area on the plasma membrane called a plaque. Desmosomes allow some give between cells, so they reduce the possibility of tearing under tension. So here's a look at some desmosomes. Cell junctions are transmembrane proteins that form tunnels that will allow small molecules to pass from cell to cell. These are very important in some of your cardiac and smooth muscle cells where the signal has to pass quickly from cell to cell. So these are gap junctions. And you can see the channels that are formed by connexons, which will allow molecules or ions to pass quickly through. Many substances must constantly move across the cell membrane. And this is where we get into the plasma membrane being selectively permeable, allowing only certain molecules to cross. And two ways that substances can cross the plasma membrane are with passive transport and active transport. And we'll talk about these different transport mechanisms, and we'll always label them as a passive transport process or an active transport process. Passive means that no energy is required, and active transport means that ATP is needed, or energy, to move something across the cell membrane. You should know the three types of passive transport. They all involve diffusion, which is just a natural movement of molecules from an area of high to low concentration, and we call this the concentration gradient. How this works is that all molecules are constantly moving in motion and they collide with each other. And in the process of diffusion, molecules will just naturally be dispersed from a high concentration area to a lower concentration area. And a great way to describe this is if we put a dye pellet or even some sugar in some water, the dye pellet will eventually dissolve and diffuse throughout the whole beaker of water, or the sugar will dissolve, or the salt. If you spray perfume, let's say in the corner of your room, eventually those perfume molecules will disperse and go from an area of high to low concentration to spread out, and that is called diffusion. The speed of diffusion is influenced by the concentration, molecular size, and temperature of the molecules, and I'll let you guys read through that. So I'm going to keep moving here, but basically how molecules that are able to passively diffuse through the membrane without any energy, so this is passive, these are lipid-soluble and nonpolar substances. Also, very small molecules can pass through, and this is just referred to as simple diffusion. If you have a larger or a non-lipid soluble or a polar molecule, they can also pass through the membrane, but with the help of a carrier molecule or a carrier protein. And we refer to this as facilitated diffusion because they're getting across the membrane with help, but it's still a passive process. Osmosis is a special name for the movement of a solvent like water and not molecules. So up until this point, diffusion and facilitated diffusion has to do with the movement of a solute particle, but osmosis is the movement of a solvent or water. Another clinical homeostatic imbalance. If a plasma membrane is damaged, substances can diffuse freely into and out of the cell, compromising or kind of getting out of whack concentration gradients. And a good example of this is in burn patients. It's so dangerous to get burned, especially thinking third-degree burns, because these patients are losing precious fluids that are coming out of their cells, proteins and ions that are just weeping from their damaged cells. So simple diffusion, some examples of things that can pass through. Again, it's small or very small polar substances like water can even pass. Yes, osmosis is an example of passive transport as well. So here's just an example of simple diffusion, molecules going from an area of high concentration to low concentration, in this case, into the cell. This happens passively with no energy expelled. Facilitated diffusion, things are transported, usually larger molecules like glucose, amino acids, and ions. They're still transported down their concentration gradient, that's passive, but they usually need a protein carrier or a water-filled channel to help them get across. Carrier-mediated facilitated diffusion, I'm not going to get into too many details on this on the test, just uses carriers to get large molecules across. And this is an example of carrier-mediated facilitated diffusion, which uses a protein carrier to get a lipid-insoluble solute or a large molecule across the membrane. We use channels with aqueous-filled cores or filled with water as transmembrane proteins to get things across. If it's a water channel that water goes through, we call it an aquaporin. You'll see that when we talk about the kidney, so just remember that word later on into the semester, like at the end. We can have leakage or gated channels. Leakage are always open, where ions are always kind of leaking in and out, and gated channels are always controlled by a chemical or electrical signal. So here's a look at diffusion through the plasma membrane, the channel-mediated facilitated diffusion. And then we get to osmosis. So this is a movement of a solvent, most often water, across the membrane, water will diffuse through the lipid bilayer or through specific water channels. And the flow occurs when water concentration is different on two sides of the membrane. So osmosis is just the flow of water across a selectively permeable membrane. We talk a little bit about osmolarity, which is just the measure of the concentration of the total number of solute particles in a solvent. Water concentration varies with the number of solute particles. So if water solute concentration goes up, water concentration goes down. And the big thing that you just need to remember is that water always moves from an area of lower solute or high water concentration to an area of low water concentration. And I'll show what I mean by that. Solutions will eventually create an equilibrium where you have the same concentration of solutes and water on both sides. But a good way of just kind of showing this is by showing this picture. So here we have kind of a U-shaped beaker, and we have a freely permeable membrane, which means that anything can pass through. So this means that water and solutes will pass through the membrane. It's permeable to both solutes and water. So at equilibrium, you'll have both sides have the same osmolarity or concentration of solutes, and the volume is unchanged. So the volume will be equal on both sides. Osmosis occurs when solutions of different osmolarities are separated by a membrane that's only permeable to water. And I'm just going to go ahead and go right to this picture because this explains it a little bit better. So here we have a U-shaped beaker, and it's permeable to water, meaning water can go through this yellow dashed line, but the solute molecules, the sugar, cannot go through. So how can we reach an equilibrium, meaning the concentration of the sugar will be equal on both sides of this membrane? Well, water has to move. So water will always move from an area of low solute concentration to high solute concentration. Or another way of saying that is water is always going to move into an area where there's more solute particles because it wants to equilibrium or equilibrate the concentration of those solutes on both sides. So what happens when water moves into an area of more sugar molecules? Now, because there's more water on the right side of this membrane, we have an equilibrium because both sides now have the same osmolarity. That just means that they have the same ratio of sugar for water molecules, so they're at equilibrium. But you'll notice in the U-shaped beaker that we needed more water on the right side of it because the right side of it had more sugar molecules. So hopefully that makes sense. Another easy way to remember this is just to remember the kind of phrase that solutes suck. So for example, the solute molecule, sugar, will suck more water onto that side because water always wants to go into the side of more solutes. The movement of water involves different pressures: hydrostatic pressure, osmotic pressure. I don't think I asked you guys questions about those pressures. Water will always try to equal out and create an equilibrium. Animal cells, and we're gonna get into tonicity now, and this has to do with osmosis, but tonicity is the ability of a solution to change the shape or tone of the cells by altering the cell's internal volume. So we're going to describe what these solutions are: isotonic solution, hypertonic solution, and hypotonic solution. And these always refer to a solution that a cell will be placed in. An isotonic solution means that it has the same osmolarity or concentration as the inside of the cell. So let's say this is a beaker. I'm going to put a cell inside my water bottle. The water is isotonic if the molarity of the cell is the same as the osmolarity of my beaker. A hypertonic solution means that the water in the beaker has a higher osmolarity, meaning there's a higher concentration of solute inside this water right now. So when I put a cell in here, water will flow out of the cell, resulting in cell shrinking, and we call that crenation. And if inside my water bottle is a hypotonic solution, it means it has a lower concentration of the cell. So if I were to place the cell into a hypotonic solution, water will flow into the cell, resulting in cell swelling, and the cell could burst. I'm going to pass over osmolarity because I won't ask you to calculate any number of moles or ion particles. But this is what I just described. And this is why it's really important to give saline solutions that are like perfect concentration of the IV or the solution that's inside the body. So let's just say in black is the type of solution, and we're putting a cell in different types of solutions. In isotonic solutions, we put a cell into an isotonic solution, meaning the concentration inside the cell is the same as the concentration of the solution. So water, you can see these arrows are describing the flow of water. Water will flow into the cell and out of the cell, but there's no net movement of water because it's completely equal. The concentration is equal. A hypertonic solution means that the solution is more concentrated. So when we put a cell in a hypertonic solution, due to what we learned in osmosis, osmosis always wants to travel. Solutes suck. Osmosis will always travel into an area of higher or greater solute concentration. So when we put a cell in a hypertonic solution, water will naturally flow out of the cell because it's by osmosis, the water is going into an area of higher concentration, into the hypertonic solution. When water leaves the cell can shrink. That's called crenation. Very bad. You don't want your cells to do that. The opposite to that is if we put a cell into a hypotonic solution. And I'm just going to emphasize the O in hypotonic solution because that's the shape the cell will make when placed into a hypotonic solution. When placed into a hypotonic solution, hypotonic means that the solution around the cell is less concentrated. So which way will water travel? Water will always want to travel into an area of higher concentration of solutes. So the water will naturally flow into the cell. It'll cause the cell to swell and possibly burst. And that's called cytolysis, also extremely bad. You should know these three solutions and what happens to cells when placed in each of these three solutions. All right, and then moving on to active transport, I'm going to just start getting through active transport, and then I might end because I want to keep these to an hour. So we'll get a little bit through active transport, and then we'll continue this on Thursday. The two major active membrane transport processes, and again, active transport means that ATP is used. So both will require ATP, active transport, vesicular transport. This will be the case to move substances across the plasma membrane if the solute's too large, not lipid soluble, or not able to move down the concentration gradient. It will require, active transport requires carrier proteins, pumps, and some carriers can transport more than one substance at a time. An antiporter transports one substance in while the other out. So we kind of are doing an anti-port system. And symport will transport two substances at the same time. Active transport always moves solutes against their concentration gradient. So think of passive transport as getting off a really crowded bus. It's easy to just get off the bus. Active transport is forcing your way into an extremely crowded bus where there's no room for you. So moving against the concentration gradient requires energy. We can have primary and secondary active transport. Primary requires energy that comes directly from ATP hydrolysis or the breaking up of those ATP bonds. And secondary active transport requires energy that's obtained indirectly from other ionic gradients. So moving on to active transport, and again, I'm going to kind of trying to focus your studying here. Primary active transport, again, breaks apart ATP to create energy. Probably the biggest one you should know, the primary active transport system that we'll talk about a lot this semester is the sodium-potassium pump. We also have other types of pumps, calcium and hydrogen, but the big one is your sodium-potassium pump. It's the most studied because it's located in all plasma membranes, but especially in excitable cells. So we'll talk a lot about your sodium-potassium pump in all nerve and muscle cells. Leakage channels located in membranes result in the leaking of sodium into the cell and leaking of potassium out of the cell. So those leakage channels happen passively. They are traveling down their concentration gradients, but the sodium-potassium pump is active because it works as an antiporter that pumps sodium out of the cell and potassium back in. And I'm going to say sodium and potassium a lot. So sodium is Na and potassium is K. So I'll probably refer to them as just sodium and potassium. So you might need to learn quickly their atomic symbols. The sodium potassium pump maintains an electrochemical gradient so that your muscle cells, your nerve cells can constantly be electrically charged to do their work as a muscle or nerve cell. So this is the sodium potassium pump, how 3 sodiums get transported out and 2 potassiums get transported in. You guys can read through the processes of this, but I won't ask you specifics on that, but you should probably know because we will eventually go through that. Secondary active transport depends on an ion gradient where energy is stored in these gradients is used indirectly to drive the transport of other solutes. I'm just going to leave it at that. Knowing what secondary active transport is important, and you don't have to know more than that. So here's secondary active transport. It's driven by the concentration gradient caused by the primary active transport. So here we have the primary active transport, the sodium potassium pump creating a gradient, meaning there's more sodium outside the cell, and secondary active transport then diffuses that gradient. As sodium diffuses back in through the membrane, it drives the glucose against its concentration gradient. So that's secondary active transport. Vesicular transport just involves transport of large particles. This requires ATP. We have endocytosis, transport into the cell, and exocytosis, transport out of the cell. Endocytosis involves formation of protein-coated vesicles. Read through endocytosis, but I'm just going to briefly talk about what it means. You'll have a protein kind of coat and engulf what it needs to ingest. So endocytosis is the cell ingesting something. And you can see this vesicle kind of forming around the ingested particles. And then those particles, the protein coat will detach, and then the vesicles inside will go on to wherever they are needing to do in that cell. So these are the step-by-steps of endocytosis, which you can read through. I won't ask you specifics about the steps in endocytosis. I just want you to know what endocytosis is. And it's an active transport process. So this does require energy. Phagocytosis, it's a type of endocytosis. It's a cell eating. So it means that solid particles will be engulfed, forming a vesicle. So phagocytosis is eating large solid particles by the cell. And that is found here. This happens a lot in your leukocytes. So some of your white blood cells will phagocytize or eat something. So we'll talk a lot about phagocytosis or phagosomes that'll actually eat to try to digest anything foreign in the body. So that's part of the immune response. And then pinocytosis is cell drinking. So the same type of idea of endocytosis, but the cell is drinking a fluid component. So this is pinocytosis. So a smaller kind of liquid component that the cell is gulping or drinking. Receptor-mediated endocytosis just means that specific molecules will bind to receptors that it needs to engulf. Some examples that use receptor-mediated endocytosis are enzymes, lipoproteins, iron, insulin. For insulin to get into your cells, it uses receptor-mediated endocytosis. So insulin will bind to specific receptors that will take it into the cell, as well as viruses, diphtheria, and cholera toxins can also be taken in this way. So this is receptor-mediated endocytosis, where the molecule will bind to a receptor, and then that will form a vesicle to take it into the cell. Exocytosis is the process where material is ejected from the cell. So think of the cell kind of vomiting or spitting things out. Usually the substance is enclosed in a secretory vesicle. The protein on the vesicle is called a V snare. It finds and hooks up with a target T snare proteins. Some things that are exocytosed in your cell, big ones are hormones and neurotransmitters, like acetylcholine, mucus, and cellular wastes are exocytosed. So this is exocytosis and the steps. Again, I won't ask you specific steps. It's fun to see. This is an actual scanning electron micrograph of about 100,000 times magnified of a cell exocytosing its contents, which is kind of cool to see. And these are the steps of each. All right, Resting membrane potential. Let me go ahead and I think I'm going to stop here. And on Thursday, we're going to finish here. Just going to check to see what slide I'm on, 101. We'll finish kind of part 2 on Thursday, and we're going to jump right in. Hopefully there will be a couple plus questions, but I'm always happy to answer your questions